Thanks, Peter, and thank you very much for having me. I'm delighted to present the Argenica story to the viewers today. So just quickly on our forward-looking disclaimer on our forward-looking statements in this presentation. So a quick company snapshot of who is Argenica Therapeutics and what are we looking to do. So our vision is really to commercialise best-in-class novel neuroprotective therapeutics, which is aimed really at reducing brain cell death following stroke and other types of brain injury. Our lead candidate to do this is ARG-007, which has shown efficacy in a number of stroke animal models and other types of brain injury models. And we're really thrilled to continue to partner with the Paraneuroscience Research Institute and the University of Western Australia, where this technology came out of. And we will partner with Linear Clinical Trial Organisation to undertake our phase 1 clinical trial. The company has a really strong IP position. We have granted patents in the EU, Japan, China, and the US, and they are 100% owned by Argenica. So just a quick company snapshot in terms of our capital structure. So we have just over 73 million shares on issue. Cash balance at the end of the last quarter was around $5.3 million with a market cap of 46.8 million at a 64-cent share price. So those funds will take us to the end of our phase 1 clinical trial. So why are we focusing on stroke and why is it so important? Stroke is a huge global issue. There's around 15 million people that suffer a stroke globally every year. So that equates to around a stroke every 19 minutes in Australia. And of these 15 million, around 5 million people die, and the large majority that survive sustain long-term disability and lasting brain damage. And it's this lasting brain damage and disability that we're hoping that ARG-007 can help. So what happens when you have a stroke? There's a reduction in blood flow to the brain, and this results in a huge amount of cell death in the brain. This equates to around 1.9 million brain cells dying per minute. There are currently no universally available drugs that protect these brain cells following stroke. And so the only treatment really is to work out what's causing that stroke. So removing the clot if it's an ischemic stroke. So how does ARG-007 work? So as I mentioned, when someone sustains a stroke, there's a huge amount of cell death in the brain. So that initial area of cell death is what's known as the infarct, and that results from that inadequate blood supply to that particular area of the brain. So initially, it only affects that part of the brain, but what happens is it sets off a cascade of cell death that proliferates out from that initial infarct area. So whilst no drug can stop that initial infarct area, what ARG-007 aims to do is to work on multiple mechanisms of action to stop that cascade of cell death that happens after that initial injury. So what we've shown in quite a number of animal models is that our drug, ARG-007, is able to slow that progression of neuronal cell death and preserve that still viable brain tissue, thereby increasing the amount of available salvageable brain tissue. So we have extensive data, as I mentioned, in animal models of stroke, but also we're looking to expand this out into traumatic brain injury and hypoxic ischemic encephalopathy. So just to give you an overview of what currently happens in stroke treatment. So when a paramedic arrives to a patient that's likely to be having a stroke, there's very little that they can do in terms of in-field triage. So the aim is really to get them to hospital as soon as possible. When they arrive at hospital, it's really important that the type of stroke that they've had is diagnosed. So whether it's an ischemic stroke, which makes up the majority of all strokes and is caused by a clot in blood vessels in the brain, or whether it's a hemorrhagic stroke, which is a bleed on the brain, because the treatments for those two types of strokes are really quite different. But all this time in going from in-field triage, in arriving at hospital, scanning, and then providing an appropriate treatment, there's rapidly increasing levels of neuronal cell death and damage in the brain, which can result in long-term functional disability. So what we're aiming to do with ARG-007 is provide it in the field by paramedics. So it's essentially stopping that cascade of cell death, buying that patient more time to get to the hospital and to get the treatments that they need, reducing that overall level of cell death and providing greater functional outcomes for these patients. What we have discovered to date is that ARG-007 can reduce the amount of brain tissue death by up to 67%, and this has been done in non-human primate animal models of stroke. And we see that that protection lasts out to 28 days. We've got preclinical studies showing that if it's given to patients that are sustaining a hemorrhagic stroke, which is the bleed on the brain, that it doesn't exacerbate that bleeding. And preliminary studies around pharmacokinetics and our toxicology studies to date have shown no adverse effects. So what are we aiming to do now as we move forward into our phase 1 clinical trial? So we're currently finalising our pharmacokinetic studies and our GLP safety and toxicology studies in animals. So these studies are required for us to move our drug into the clinic, into healthy volunteers. We've kicked off our pre-study activities for our clinical trial, and we aim to have our phase 1 ethics submission in Q2 this calendar year, so not too far away. So just to give you a bit of an overview of what we're aiming to do in the phase 1 clinical trial, it is really a trial to assess the safety of our drug in healthy volunteers. So we administer different doses of the drug and deem what is the ideal safe dose when we want to progress into our phase 2 clinical trial in actual stroke patients. So just a quick snapshot of the trial design. It's actually quite a short trial. There will be 4 cohorts, each receiving a different dose of ARG-007, with 8 participants in each cohort. So we'll have 6 receiving the drug and 2 receiving a placebo. The patients come into the linear clinical trial facility and they receive the drug by intravenous infusion. They stay over for around 48 hours and a number of pathology samples and data is collected over that 48 hours, really to give us an idea of whether there's been any adverse reactions and to show that the drug is safe. These volunteers come back at day 8 and more pathology samples and data is collected. Then we move on to the next cohort, which is a slightly elevated dose of the drug. So following a phase 1 study, once we know that our drug is safe and well tolerated in healthy volunteers, we'll look to progress that into a phase 2 study in stroke patients. But the fantastic thing is that once we know the drug is safe, we can actually progress the drug into phase 2 studies in other indications. So as I mentioned previously, HIE and TBI are 2 areas that we already have preclinical data on that we're looking to progress, as well as surgically induced stroke. So that's stroke sustained during surgeries such as aneurysm repair and cardiac surgery. So we have supported by a fantastic team with experience in not only capital markets, neuroscience and biotechnology. So Jeff Pocock, our chairman, who is currently non-exec director on Envision Medical Devices and previous co-founder and former managing director of Hazer Group. From a management perspective, I'm supported by Dr. Samantha South and Emma Walden. So just a quick snapshot on our investment highlights. We have a really clear pathway to move into our phase 1 clinical trials in Q2 this year. We are addressing a huge unmet medical need in stroke, but also moving into TBI and traumatic brain injury. We're really fortunate in that this drug has close to 10 years of preclinical data supporting it to show the efficacy and the safety of it as we move into the clinic. We've got an exceptional management team and board to help us get there, and really exciting near-term catalysts as we get all our preclinical data to move into our phase 1 clinical trial and then beyond into various phase 2 clinical trials. So thank you very much for your time today, and I welcome any questions. Thanks.
Thanks, Liz. And for those watching, make sure you get your questions in as soon as you can. Liz, I am interested that you called the drug ARG-007. Is that because it's licensed to cure?
I actually haven't heard that one before. That's very good. I'm going to use that in the future. Yes. So it's actually an iteration. So we started with a smaller peptide, so one that had 12 amino acids, so 12 arginines in a row. And so it was actually just an iteration. It just happened that the most efficacious drug was the 7th one, essentially, in the line that we tried. So yes, but it works well, I think.
Yeah, okay. What is the impact on someone who's got a stroke and has gone through the hospital system, and then is able to access the drug? What happens to their cell repair or limiting of destruction?
So ideally, I mean, ultimately what we want to do is to be able to give this drug in the ambulance, so by paramedics. So when someone presents with stroke-like symptoms, which might be sort of that face paralysis, droopy mouth, unable to speak, ideally we would give that drug. So we've already gone through the process in clinical trials to show that it's safe, that it's effective. So we would give that drug in the ambulance, and essentially what that's doing for that patient, it's stopping that cascade of cell death. So it's essentially buying them time to get to the hospital. So when they arrive at hospital, they will be rushed in, there'll be diagnoses to the type of stroke that they're having. The neurointerventional radiologist will go and have a look at those scans, and then they'll work out the type of treatment. So can they remove the clot surgically, or will they administer a drug that can dissolve the clot? Or is the clot in such a sort of place that nothing can be done, essentially? So really what our drug is, it's protecting the brain cells essentially during that whole process.
Yeah. So I would presume that the time when you get the biggest bang for your buck in terms of potential approval will be in phase 2, when you have these surgically induced strokes, where you've got doctors knowing there's something going wrong, and they then can apply ARG-007. That's when it would be the best test, wouldn't it?
Yeah, absolutely. And we're definitely looking down that path. So when you have aneurysm repair or valve repair, there's a higher risk of actually sustaining a stroke. So a little bit of something breaking off and a clot traveling to your brain. So we could almost look at it prophylactically. Could we give it to patients before they undergo these surgeries? So we're definitely exploring those possibilities in clinical trials.
Okay. Question from Rob Matthews. Hi, Liz. When you complete your ethics submission, approximately how long does it take for approval?
So the ethics will go to Belbury, which is a human research ethics committee. They turn it around pretty quickly. So they've got an average 20-day turnaround. So from ethics submission to ethics approval, it's pretty quick. And then as soon as we get that ethics approval, we start that patient recruitment immediately to get into that phase 1 clinical trial. So that process is quick. Yeah.
Question from Robert King. Have you been working with the FDA on the design of the phase 2 trial to ensure all the required data is being gathered?
Absolutely. So I think we can see this time and time again with biotech companies where they don't consult the FDA early enough. So we have FDA consultants based in California that are helping with our phase 2 trial design at the moment, and we'll take that to the FDA. So even if we decide to do that entire phase 2 in Australia, we'll make sure that we have that input from the FDA so that we don't come unstuck as we ask for our IND for phase 3.
Okay, Liz, good luck with that. It's a fantastic product you're trying to bring to market. Let's hope it works, and good luck with the company going forward.
Thank you so much. Thanks for having me.